on
Obsessive compulsive disorder, better known as OCD, is a mental disorder where you have thoughts, which are obsessions, and rituals, which are compulsions, repeatedly and uncontrollably to the point where they interfere with your everyday life. OCD is a fairly common mental disorder that is found in around 2% of the general population. The development of OCD can stem from many different pathways and origins, ranging from the environmental factors to genetic inheritance. Many of the genes that play a role in OCD are related to serotonin levels or serotonin transportation. The SLC6A4 and the TPH2 genes both play roles in the regulation of serotonin levels, and both have genetic variations that impact the development of OCD. The COMT and FMR1 genes are also both associated with OCD. The COMT gene codes for proteins that break down neurotransmitters, and the FMR1 gene codes for a protein that plays a role in the development of connections between brain cells.
Our SNP chip test specifically focuses on two SNPs, RS25532 and RS4570625. The RS25532 SNP is associated with changes to a regulatory portion of the SLC6A4 gene. The SNP has a CT promoter sequence variant, of which the presence of the C allele has been shown to be strongly linked to an omnibus haplotype that is connected to the development of OCD. The general function of this gene is to produce a protein that collects serotonin in the neurons, which is important in many psychological disorders like OCD. The RS4570625 SNP has been shown to affect the brain's ability to process negative emotions. RS4570625 is located on the TPH2 gene, which affects serotonin synthesis. The RS4570625 SNP has two alleles, G and T, of which the G allele has a 74.6% correspondence, and the T allele has a 25.4% correspondence with the development of early onset OCD. Research suggests that both of these SNPs have clear associations with obsessive compulsive disorder and other psychological disorders. Our ability to predict the onset and severity of psychological disorders such as OCD could be improved by testing for these specific polymorphisms on a SNP chip test.
As we previously mentioned, the two major SNPs that we will be testing for are the RS25532 and RS4570625 SNPs. The first SNP we discussed was the RS25532 SNP, which has two alleles, C and T. There has been shown to be a significant increase in the frequency of a haplotype associated with the development of OCD when the C allele is present. Testing for this SNP actually allows for us to get a bigger picture of your body's genetic information than just this specific variation. It allows us to get a better sense of whether you possess the haplotype associated with OCD. The other SNP that we discussed earlier was the RS4570625 SNP, of which the G allele has a 74.6% correspondence, and the T allele has a 25.4% correspondence with the development of early onset OCD. The benefit of our SNP chip test is that it allows for us to test for both alleles, which can give us an even more detailed genotype analysis of your specific genetics, as shown by the table below. The GG genotype has a 53.5% correspondence, the GT genotype has a 42.3% correspondence, and the TT genotype has only a 4.2% correspondence. The biggest benefit of our SNP chip test is that it allows for you, the consumer, to get a more defined and empirical knowledge of your specific genetic situation. This allows you to make more educated decisions on how you want to structure your life and if you want to take any preventative or preemptive measures that are based on scientific facts.
No matter the potential benefits of our tests, there are still some potential limitations and considerations that you should understand. First, you should look at the cost and whether a costly genetic test is actually worth it given your knowledge of your genetic history. You also should understand how we came about the information that informs the production of our SNP chip test. We have analyzed many studies that research genetic variations related to OCD. We have chosen several SNPs that have been shown to have many changes in the phenotype of OCD. However, these might not provide the whole genetic picture for a complex multifactorial trait like OCD. This can be further shown by the HTR2A gene, which had been linked to OCD in the past, but recent studies have found no connection between the gene and OCD. This could have potential ramifications toward the genes that we test for our SNP chip test.
There are also some ethical considerations of our SNP chip test for OCD. The most obvious is the potential emotional effect that testing positive for genetic variation related to OCD can have. There are two potential outcomes that could come from that result. One, you could become very worried and anxious about the possibility of the disease revealing itself, which can cause severe emotional and social distress. However, the other potential reaction could be a new focus on appreciating and enjoying the life you have before the effects of OCD start to appear. Additionally, one ethical concern related to OCD genetic testing is that of confidentiality. Do the tests provided remain entirely confidential? If not, could the results of genetic testing be reported to insurance companies or employers? This is definitely of concern when considering whether or not to be tested. Finally, predicting the onset of genetic disorders, such as obsessive compulsive disorder, does not necessarily allow for prevention or mitigation. Some even argue that the presence of certain genetic variations is not enough to warrant testing unless the individual has an obvious family history.